Hello, everyone, and welcome to today's webinar hosted by Jen. Our topic today is reducing cost and risk in lentiviral manufacturing with the LV Edge packaging system. Today's webinar is sponsored by Asimov. Thank you so much for being here. I'm Uduak Thomas, Senior Editor with Jen, and it is my pleasure to be your host for today's webinar. Our speaker is Mark Stockdale. He's the Senior Director of Strategic Alliances at Asimov. We'll get to his presentation in just a moment, but before we do that, let's get some background on the topic of the day. In the manufacturing of lentivirus vectors for cell and gene therapy applications, GMP-grade plasmids account for a significant proportion of the raw material costs. Procuring these plasmids also introduces supply chain complexity, which can impact both timelines and product variability. In this Gen webinar, our speaker will discuss the challenges and bottlenecks that are associated with lentiviral manufacture and introduce the LV Edge packaging system, which minimizes GMP plasmid cost, process complexity, and supply chain risk by stably integrating viral genes into the host cell. Some of the key topics that will be covered in this webinar include how the LV Edge packaging system enables tunable transgene expression in transduced cells, how a clonal suspension adapted packaging cell line reduces the need for GMP plasmids, how model-guided DNA design can optimize transgene expression and lentiviral titer, and lastly, how to access the system to reduce cost and risk in the design and manufacture of novel therapeutics. Before we begin, a reminder that we would love to take your questions following our main presentation for the day. So at any time during the presentation, feel free to enter your questions in the Ask a Question panel on your screen. All right, let's get started.
Thank you. It is a pleasure to be able to share Asimov's latest developments in lentiviral production. The structure for today's talk is to provide you all an overview of Asimov, then provide you exciting data on our two LV Edge systems, with a final summary at the end. By the end of the talk, I hope you will see how Asimov's quantitative synthetic biology systems have been applied to generate a reliable system for manufacture of lentivirus vectors. And the highlights for today's talk will introduce the packaging and producer cell lines with the stable integration of viral components, show you some stellar performance data of greater than e to the 8 transduction units per mil for therapeutic transgenes, and how by using this packaging system, COGS might be reduced and we de-risk the supply chain. I'll also provide you a sneak peek of the LV Edge producer system. To call out at the start, Asimov does not do CDMO services. We're not a contract manufacturer. We provide this technology to innovator companies, our customers, and also CDMO partners to perform that production on your behalf. So let's get started. We unify software and biology for the production of therapeutics. The Asimov technology platform is formed from host cell lines like HEC293 and CHO. We have a library of curated genetic parts. We build data-driven models around process and genetic design. And to understand all of this complexity, we've built our own software for the computer-aided design of biology called kernel. Our customers access this technology directly through tech-out licensing. This is where we drop the technology into your lab, and then myself or my colleagues support you as you develop this technology and use it for your production of lentivirus. Alternatively, customers can initiate services with us for cell line development services, and that's the particular case for the lentiviral producer cell line and our biologics CHO cell line. We also work closely with CMOs for scale-up. We have 3 pillars of activity: biologics, cell and gene therapy, and RNA therapy. We provide technology to support these areas. We have a mature product for biologics production called CHO Edge. We have nascent applications around mRNA therapeutics, and some of this technology includes tissue-specific expression. For today, I will focus on our advanced therapy applications for cell and gene therapy, in particular LV Edge, and how we developed a new production system for lentiviral particles. Transient production has been the mainstay for lentivirus production. It allows that speed to clinic, but it has limitations. There are many challenges we see doing transient transfections at scale, and that topping out around 2,000 litres to 1,000 litre bioreactors is one of those. Some new lipids may address that going forward, but there's still the cost of the GMP plasmids. To address these challenges with transient production, Asimov has developed two options. Our packaging cell line, which has similar fast development times to clinic with a reduced cost and complexity, and then our producer system, developed for larger indications to allow scalability while maintaining reduced cost of goods even further. So in summary, we have transient speed, but has limitations. Asimov's packaging, similar development times to transient, which reduces complexity and cost, but still limited by that scale. And then Asimov's producer system for scalability, but limited by development timelines. Let me now introduce our semi-stable system, LV Edge packaging system. The Asimov LV Edge packaging system has three cornerstones: the packaging cell line, DNA design, optimization for LV, refinement of the platform process with modeling. I will firstly introduce these areas, then delve deeper into some of the background data. Firstly, the packaging cell line. On the left shows the provenance for our host cell line. Our team in Boston, USA, sourced the adherent HEC293T cell line from ECACC. They engineered this cell line to incorporate the helper viral genes for Lenti, Gagpol, Rev, VSV, G. These genes have been stably integrated into the HEC293T genome. To overcome the toxicity of these viral genes, we have inducible control of their expression. This also shows improved stability. Important for GMP use is having a clonally derived cell line to ensure consistent future supply of the therapeutic lentivirus. So the stable cell line was cloned out and suspension adapted to create a clonally derived packaging cell line. And this process is fully documented in terms of its creation. We have a research use cell line ready to ship, and the cell line coming out in mid-2024, which will be GMP banked. Secondly, I will show you how we can help with the DNA design of payloads. Our CAD software kernel was used for sequence optimization and designing of the transfer plasmid. Kernel provides a visual representation of the transfer plasmid, and within kernel, we can use it to design the contents of that transfer plasmid, adjust promoters, and in particular, we can optimize the sequence based on internal development work that we've done within Asimov in terms of improving that sequence. So looking at that data, taking 3 commercially CAR-expressing lentiviruses, we took the original described DNA sequences, shown there at the top, and we applied our sequence optimization. And here's the data in terms of the functional titer shown on the right-hand side. Across the 3 vectors, we see a 4-fold to 17-fold gain in functional titer compared to the original described sequence at the top. We measure functional titer by flow cytometry. I will describe this assay in further detail later in the talk. This was an interesting observation to see such a large gain based on our internal Asimov sequence optimizer. So we also benchmarked against other sequence optimization strategies from third parties. These companies show varying improvements depending on the original sequence, but the Asimov sequence optimizer parameters give consistent improvements for these different CARs shown here. We are going to explore this further but the important take-home message here is that we do have an ability to improve packaging and production. So before you lock in your sequence for your transgene, it's always good to come out, reach out to us, and we'll see if we can optimize it further for you. Lastly, our platform process has been built to be off the shelf. For our off-the-shelf process, we spent much effort to build an optimized process that can be easily transferred to other laboratories. Our team has looked at different reagents, whether transfection reagents or cell culture media. We run these tests at different scales. To make the transfer of the technology as easy as possible, we always keep a line of sight on the ultimate goal to scale these processes up. There have been many experiments in the lab, and also our computational biologists have helped guide and build models to help with this optimization. Here on the right, we have a simplified experiment looking at two-dimensional space of process A versus process B. Feeding the model with training data, we start to see the greater impact of process B for titer. We want to expand this capability to help fit our system into new processes that benefit production and ultimately the patient. So that covers the top-level introduction to the LV Edge packaging system we've just launched, a semi-stable method to generate lentiviral particles. So we have the packaging cell line, a clonal and suspension adapted, stable integration of viral genes. We have sequence optimization of payload, experimentally validated transfer vectors. And then for the platform process, we have robust protocols and expert technical support to guide you through the establishment of this technology within your own labs or at a CMO. I will now delve deeper into the process and our supporting data. So how do we use the packaging cell line and how have we generated the data shown today? At Asimov, we take the sequence, we perform the optimization in kernel, we build that transfer plasmid with your gene of interest. Then the gene of interest is transfected into the LV Edge packaging cell line. The viral genes are induced and the packaging cell line drives the production of the lentivirus particles. Here shown on the right-hand side, we have some supporting data with 3 clinically relevant vectors where we achieve 8 to the 7 transduction units per mil for anti-BCMA, going up to 1.5 e to the 8 transduction units for Chimera. A very respectable titer indeed, especially considering this is unconcentrated harvest. The process is very similar to transient with the benefits of the speed to clinic. Importantly, there is only the need to transfect 1 plasmid. What I've shown you has been functional titer. It's probably important just at this stage to delve deeper into our biological assay, into how we determine functional titer. For developing this platform, we wanted a biological assay that could show us the impact of our changes to the genetic system and the process and give us a rapid readout. And some of those considerations about do we use unconcentrated and looking at developing a facts-based method gives us the speed to address those changes and to iterate quickly. Of course, that may be different between different labs in terms of how labs assess functional titer, but today I'll just show a little bit more about how we do it here at Asimov. We have our cells producing lentivirus. To understand that functional titer of the virus, we harvest supernatant and add that to our model HEC293 cell line that we use for the biological assay. We add varying amounts of the harvest supernatant, and at the higher amounts we confirm that we have virus present, and we see this clear shift in terms of the stained population that's expressing the CAR. But for the true analysis for the functional titer, we then look at a sweet spot for analysis, and in particular for this example shown here on the right, we use 1 microliter. So we infect The infected cells are stained for the CAR, and then we determine the number of infected cells based on that staining for the CAR being expressed from the integrated lentiviral vector. The analysis of flow data took us some iterations to find the best method, and we landed on the Gaussian model. One of the benefits of Asimov is that we have a melting pot of different perspectives and skill sets to really get the best out of the assays and the data, and to really understand what we're doing. On the right, we'll delve into this in a little bit more data. So we took the 1 microliter sample, and we see here the original sequence has this slight movement of population towards the positively stained. For the mean expression, the shift to the right, we have a 6-fold change between the original sequence shown here and then the Asimov optimized sequence. More CAR is being expressed in the infected cells. This could be really considered a measure of potency. Then the percentage of transduced cells, the number of cells under the curve, a measure of productivity, we have an increase from 47% to 60% with the optimized sequence performed at Asimov. So our transduced cells are expressing more CAR, and with our capabilities, we can dial that up or dial down that expression based on the promoters that we use for that, for driving that transgene. So we can tune this expression up or down based on the clinical need. And we have this ability that the functional titer is also improving as well. Important for us observing this result in a different cell line. So taking a clinically relevant cell line is a second readout for our functional titer. And we also perform this in-house at Asimov, our Boston lab. So we developed an internal protocol looking at establishing the functional titer within primary T cells. So the process here shown on the left-hand side, we process the primary T cells for CD4 positive and CD8 positive populations and freeze. For each assay, we activate, then transduce. For the transduction, we use frozen lentivirus, which we know impacts the functional titer because this is unconcentrated virus again. So sometimes there's a reduction in the functional virions within there. Staining for positive populations of the CAR, we see functional titers in the order of mid-E to the 6 transduction units per mil, as we might expect for unconcentrated and frozen samples. Also, we see differences between the cells derived from different donors. And the importance here again is when comparing the sequence optimized version here in the lighter pink compared to the original sequence for Yescarta, we observe a 15-fold improvement in that functional titer. In the technology transfer, it will be important for us to understand how people do their own functional titer assay for benchmarking purposes. We are aware of different methods out there. And this is part of my role and my team's role to really support that, and we look forward to having those conversations. Moving away from transient system to packaging, the benefit will hopefully be lot-to-lot consistency. But an important factor for consistency is the stability of the LV packaging system and how that might impact functional titer, and then also any potential genetic instability. So we performed that assay at Asimov to delve into that deeper. So we cultured the cells for 60 generations. From week 1 of culture for functional titer by performing the transient production at the start and at the end, we only see a CV of 2%. Looking at genetic stability, again, from this point to this point, we see this slight shift in the overall GAG, but this is a slight drift of only 5% CV. And this could be inherent in terms of the assays that we're performing. So overall, we have a very robust, stable cell line. In terms of the functional titer, there's insignificant change from within those 60 generations. And 60 generations really could be an overkill when we look at clinical supply. There's plenty of time to revive and bulk up this cell line for the transfection to even meet those larger scales above 200 liters. But yeah, the take home here is we see long-term stability of expression and the genetics of the packaging cell line. That brings me to the end of my presentation for the packaging cell line. As we discussed earlier, the packaging cell line has limitations for scale due to the need for transfections. So now I'm going to look at how Asimov's addressed that scalability issue and give you a sneak peek on the development of the LV Edge producer system. It is built on the capability to deliver a stable cell line expressing lentiviral vector in 4 months. We have a well-kitted out lab in Boston with excellent experts, people, instrumentation there to complete the cell line development for LV Edge producer. To delve into the process in terms of how we create the producer cell line, we take our HEC293 cell line and transfect all the necessary components for expression of the LV vector, the VSVG, the GAG pol, the REV, and then the gene of interest. The helper genes are under inducible control of expression to minimize the impact on the host cell line, so we don't see that toxicity of those viral genes. The stable pool is generated via transposase-mediated integration of the helper genes, and we have selection with 4 antibiotics. For each campaign, we generate multiple pools. The process is carried out in 2D culture in the presence of serum. And then we, in particular with the serum, the source of that serum is well documented, so it's very low risk in terms of the impact for TSE, BSE risks. It's took us some time as we explored the benefits of adherent versus suspension at transfection stage. The adherent offered those advantages and was the best route forward in terms of creating the stable cell line. But we acknowledge that there's risk there, so we've de-risked that step by minimizing the time in the serum and sourcing serum from reputable suppliers. The adherent stable pools are then suspension adapted into chemically defined medium. We create a suspension cell line suitable for large-scale clinical production. Surprisingly, or maybe unsurprisingly, we see a gain in titer even at this stage, where the stable pool, we start to hit those low to mid E⁷ transduction units per mil. And a reminder again, this is unconcentrated virus, so a respectable bump again for a stable producer pool at this stage. On the left, this is our process now as we take that stable pool and start to generate clonal populations. We single cell print from the top pools, confirm clonal derivation using imaging with the cell metric system. And then with those established clones, we perform a proprietary high throughput screening method to take forward the top clones. We then expand those clones, do a production run, and measure titers by a transduction assay. At this stage, we then focus on those top clones. On the right, for 2 representative LV vectors, 2 CARs, the pool in the light shows around 1 E⁸ transduction units per mil. And then for those top 12 clones derived from each of those pools, we see that 3-fold to greater than 4-fold improvement in titers depending on the different CARs. And really to see this in a stable producer setting is something that we're very excited at Asimov about. And we've seen this constant improvement in productivity. Again, this is done in HEC293 cells. So the next step for us was to confirm these titers within primary T cells, similar to what we did in packaging. So we're selecting clones that are ultimately going into the clinic. So we want to be sure that we have representative titer assessment. So taking those primary T cells with a similar methodology from what I've shown before, we determine functional titer from the unconcentrated virus. The same process was repeated as I've shown earlier with 3 donors. We then determine the titer from the top 5 clones for 2 CARs. On the right, we see that for clone ranking, the correlation between HEC-293 functional titer and the T cell is very strong and reliable. There is variability that you often see between T cell donors, but the data is still pretty tight, especially once you get to those high titer values as well. In terms of the absolute titer that we see in T cells, it is in the mid E6 to mid E7 transduction units. And we see this as what we would expect as well, based on how we've developed this assay, and it is consistent run to run. And it'd be interesting to see this translate to other labs as well as we take this technology out. So far, we've been assessing the clone titer in shake flask. The next step is to take them into a more bioreactor-like environment. Shown here is a scale up into amber. Using our model-guided approach, we can ensure clones are as good, if not better. I remember from a few years ago, amber being really difficult with suspension HEC-293s. I think here we've really cracked it, and we take these top clones, and we see a trend for increased production on that amber process in purple. So with those 3 clones, we see a very nice boost going up to those E9 or high E8 transduction units per mil. Again, important, this data is hot off the press, and this study is still ongoing within the lab with a target of 60 generations. We performed a stability study, which is necessary. So we've taken 5 stable expressing clones for 2 different lentiviruses. We determined the stability of the titers for 28 generations. And we're going to expand this out to 60 generations and expect that data coming soon. But the data is very tight for all of the clones expressing 2 clinically relevant CAR LV vectors. The CVs are low single digit. Any clonal variation we see is perfectly fine. I mean, regulatory agencies need reassurance of future supply from clonally derived cell lines expressing LV, and this data stacks up very nicely to support that. Again, this kind of brings me now to the end of our stable cell line. If you're interested in this exciting technology, please do reach out. And we've got slots coming up later in the summer for a stable producer cell line development. I will bring everything together now and summarize the top 2 systems shown here. As we apply the Asimov technology at each step, shown here is a case study with Chimera, we get a stepwise improvement for applying our technology. So initially we have that 5x-fold improvement in transient setting just by the application of our sequence optimization. Then moving to our packaging cell line plus the sequence optimization, we get comparable functional titers. And then moving forward to the scalable producer system, that we create a clone that can be used for future production of lentivirus. We get that, again, 7-fold improvement in titers with the ability to easily scale that for future therapeutic applications. I hope I've convinced you today that Asimov has a number of technologies that can be applied and also open up new therapeutic options for diseases that transient lentivirus production cannot meet today. In terms of our LV Edge system, we have the packaging cell line with unmatched production control and quality consistency. We have sequence optimization to increase potency of the therapeutics. Our in-house specialized team can help design and build DNA for the gene of interest plasmids. And we have a platform process that allows us to scale and is ready to transfer into other labs as required. We have future developments. We'll continue to curate more parts and expand our genetic simulator for LV applications. We have the producer cell line developments that I've shown today. We're starting to take bookings, and that provides scalability on demand as the customer needs dictate or the patient needs dictate. And we're further developing our model-guided approach to fit your needs, whether it is a new transfection reagent or 1,000 liter scale or different gene of interest that impacts the process itself. So thank you for your time. That brings me to the end of the presentation. I'm happy to move on to the Q&A session. Thank you.
Thank you so much for that superb talk. In a few moments, we'll get started with the live Q&A session. So if you haven't done so already, now is the time to go ahead and submit your questions for our speaker. Before we begin the Q&A, a quick reminder that this webinar will be available to view on demand in the next 24 to 48 hours. So please keep an eye on your email, and please do share the link with your colleagues who might not have been able to tune in today. Okay, we're going to start with your questions. Just give us a couple of moments to transition to the Q&A. We'll see you shortly. And a very warm welcome to the live Q&A for this webinar. Thank you so much for joining us. Mark is here and ready to answer your questions. So just a quick reminder before we get started, if you do have a question, please go ahead and enter it in the Ask a Question panel on your screen. We're going to be taking questions live for the next several minutes. Mark, thanks so much for being here and for that great presentation.
Hi, thanks so much. It's great to be here and part of this webinar.
Absolutely, absolutely. All right, I do have a few questions already for you, so I'm just going to go ahead and jump right in. First question I have, with your approach for integrating viral helper genes, do you have any concerns on replication-competent lentivirus?
Oh yes, it's always top of your mind that we need to address replication-competent lentivirus. Is a great question and it's pivotal for lentivirus production. So from our approach, we mitigate replication-competent lentivirus through designs of our system, and also we've confirmed it through performing an assay as well for looking for that replication competency. So in terms of the design, we have several key factors which influence the frequency of recombination events, where we take that into consideration in the design of our LV edge system, both for the packaging and the producer cell line. So we try to reduce the homology between the vector and the helper sequences, and then homology as well is reduced between vector and helper sequences compared to the cellular DNA, so we don't get any combination events there. And then also within the helper sequences, we divide those up to more than one expression cassette to increase the number of recombination events required to generate that RCL. And then the integration of the viral elements as well are located in a number of places across the genome, so it's not one single loci. So hence that reduces the likelihood as well. But I mean, importantly, it's the proof in the pudding. So we've tested for replication competency with the 3 clinically relevant molecules that we presented today in the packaging cell line. And those samples have been cleared. So we use an external company who've developed an assay according to FDA guidance. And for that assay, we take the viral soup, transduced cells at 5 MOI, and then those transduced cells were passaged, RNA extracted, and then running a qPCR assay to look for any presence of the virus. And if any recombination had occurred during that transduction, and allowed replication virus, it would have been detected, and in our case, it was not. So yeah, hopefully that alleviates any concerns from our audience today.
Excellent, thank you. Next question I have, do you have any theories around how your sequence optimization increases titers?
Yes, yes, good. I mean, we've designed the sequence optimizer based on a number of certain parameters to improve packaging of RNA. But I hope you appreciate that I can't go into all of the details. Some of it is trade secrets. But it's also good to note that we're currently exploring additional hypotheses to further enhance the impact of our sequence optimizer. And that's through the enhancement of titers and potency in a modular way as well as we go forward.
Excellent, excellent. Next question I have you evaluated product variability across the different manufacturing platforms? So transient versus packaging versus producer.
Yes, another great question. Indeed, yeah, stable manufacturing platforms, we kind of see it should provide that consistency in manufacturing. And therefore a more consistent viral product at the end. So there'll be less inter-batch variability. So we believe product quality and the CQA for lentivirus is a gap in the industry, and we want to help raise that standard, raise that bar in the industry. So we've seen indications of reduced variability in terms of key productivity metrics, and we're further confirming those results. Additionally, we're evaluating both viral integrity and integrated payload through NGS to further confirm the quality benefits of these platforms, particularly the packaging and the producer. So that analysis is ongoing, and I hope to communicate those results later this half. Importantly, these methods are not common practice in the industry. I suppose we see that the regulatory agencies are probably interested in advancing them, so we hope to be charging that change as well. Thank you.
Excellent, excellent. Thank you. And just to remind our audience, we're taking questions live. So if you do have a question, please do go ahead and enter it in the Ask a Question panel on your screen. We'll do our best to get through as many questions as we can in the time that we have left. So please get those questions in. And thank you to those who've already sent their questions. All right, the next one I have for you, Mark. The titer results you show in the presentation are calculated through flow cytometry. So have you confirmed your results via qPCR?
Yes, correct. So we transduce those HEC293 T cells, stain with CAR-specific antibodies, and measure the expression with flow. So yeah, there are other means and other ways of doing that, but we have a very robust assay. We further validated by using Gaussian mixture model to ensure robust results. Regarding integration copy number, titer confirmation, via means like qPCR, we have confirmed results via HEC293T transduction, followed by a qPCR method. We're an external vendor, and we see comparable results with this methodology that we use in-house at Asimov.
Excellent, thank you. So this question, I'd actually like to reduce the amount of CAR expressed in my target cell. Would this be possible without impacting the titer?
Yeah, that falls directly in our wheelhouse, really. So great thing about Asimov is that we have a number of molecular biological techniques at our disposal to allow tuning of CAR expression to a target level independently of its DNA sequence. So it's something that we take on board in that design phase as we're building that transfer plasmid and build that into what's required for the patient need.
Okay, excellent. Next question, what was the reason for selecting HEC293 T cells for transduction? I think you might have started to touch on this a little bit.
Yeah, I mean, it's an established cell line for production. It has all the components that we require for lentivirus production as well, in terms of the T antigen. So that's why we started there. And in terms of the stock of HEC293Ts, they are established stock as well that have been used elsewhere in the industry. So they have a regulatory track record as well in this space.
Excellent, excellent. Next question I have, what's the impact of such a high titer, so E8 to E9 TU per ml, on the DSP scalable purification and aggregation of LVV?
Yeah, I mean, this is probably less of an area that I have knowledge about, but I mean, the high TU per mil, the transduction units per mil, yeah, and the bio, I suppose the potential instability of lentivirus as well. It could be an area that we're really happy to explore with our customers, and also with those other companies who are developing purification systems for lentivirus as well. So we haven't seen any impact so far, but this is an area that we want to sort of delve into deeper as well.
Excellent, excellent. What is the inducible system that you use for your production cell line?
I can't probably go into that detail today on this call, but for that person, yeah, please feel free to reach out and we can sort of discuss that in greater detail with you.
Excellent, excellent. Mark, thank you so. Oh, we got one more question popping in. My apologies. I'll ask this one. How did you look at the on-off switch CAR T approach as compared to conventional gene therapy approaches, or to a conventional
Yeah,
gene therapy approach, excuse me.
I'm not quite sure I understand the question, I'm afraid. So, um, if that is particularly around the induction of viral, yeah, we have an inducible system for viral vector production, and that creates the lentivirus particles that are then used to transfer the CAR over to the target cell line. In terms of getting an on and off switch for the actual CAR therapy, yeah, that's an area that we can look at because we have those capabilities in house in terms of the genetic parts that we have. But yeah, in terms of the question, I don't know if I can fully answer that. So I'd be more than willing for that person, yeah, to reach out. We can delve into that deeper.
Absolutely. And definitely, if you have, if for this individual, if you do have, if you'd like to clarify your question, you can feel free to follow up directly with Asimov and they can help you more directly after the webinar. So please do follow up with Asimov on the website at asimov.com/contact. All right. And they can follow up with you. We have another question that's come up. How much is the infectivity across the GOIs?
That is another area that we're delving into currently. So we're using external vendors to do some of the assessment there. But what we've seen so far is the infectivity looks to be where you would expect for a lentivirus system. So there's been no concerns there. And there's possibility as well that we see a nudge when we're looking at our systems compared to others. But that's something that we'll be able to share in the future.
Excellent. Excellent. Mark, thank you so much. That is all the time we have for today's webinar. Thank you to our audience for being here and for asking such great questions. As mentioned, this webinar will be available to view on demand very shortly. Please keep an eye on your email. In the next 24 to 48 hours, you should get a link that will take you to the presentation as well as to this live Q&A session so you can watch it again, or you can also share it with your colleagues who might not have been able to join us live today. Mark, once again, thank you so much for such an interesting and informative presentation.
And thank you for running today's webinar, and thank you to the audience for your attention as well.
Absolutely. And I'd also like to thank our sponsor, Asimov, for sponsoring this event. And a special shout out to the superb Gen Multimedia Group for hosting. So we have Leanna Jabs, David Mosley, and Hannah Turner. Thank you. And once again, thank you to you, our audience, for being here today. For everyone at Gen, I'm Uduak Thomas. Thank you and goodbye for now.